isolation and characterization of bacteria found in oil-contaminated soil by Sheila Joy Bidamian and Shane P. San Pablo, SP in Microbiology. As industrialization develops, petroleum hydrocarbons become a superior prospective cause of pollutants in the water and soil environment. Hydrocarbons are the major component of pollution. It can be found in soil, water, and air. It can enter water through direct spills or from a spill originally occurring on land and subsequently reaching water bodies through the effects of wind, rain, surface or subsurface flow. The Pandacan Oil Depot, which is located beside the Pasig River, has operated since 1914 to 2012. Processing waste and petrochemical waste. This study aims to isolate bacteria from oil-contaminated soil from Pasig River Pericommute PUP station and also test if this research also tests if certain bacteria found in oil-contaminated soil can grow in nutrient agar alone. For the methodology,
soil samples were collected at the shoreline of the Pasig River near PUP Linear Park. The samples were taken down to 10 centimeters depth. Sterile containers and stainless steel trowel were used to gather samples to avoid contamination. Approximately 500 grams were collected for the two samples. For the isolation of sample, 1 gram of the sample were suspended in 100 ml of sterile distilled water then agitated manually. Aliquots of 0.1 ml from each dilution were spread over the surface of nutrient agar plates. For the bacterial identification, the morphological characterization of each isolate was first performed, including color, size, colony characteristics, and gram staining was performed for each isolate, and the ability of certain isolates to grow at room temperature were also investigated.
For the results and discussion, the nutrient agar plate showed significant bacterial colony growth, which indicates that bacteria are living and growing abundantly in oil-contaminated soil samples. The clear appearance of any plates before isolation became opaque, and several colonies appeared after 48 to 72 hours of incubation. The colonies produced creamy to brownish in color and some way black due to the residues of the aliquots from the agitated soil sample. Colony character. Here's the table showing the results from our isolation. From sample A, size is small, sample B large, shape and arrangement both are round and in shape, opacity both are opaque, margin, sample A is regular, sample B is irregular, elevation and pigment, both are slightly raised and creamy to brownish in color.
From the table above, in comparison of morphology and microscopy from other studies, the bacteria species can be identified as Bacillus subtilis. The main habitat of endospore-forming Bacillus organism is the soil. Likewise, Bacillus subtilis is most commonly found in soil environments and on plant undergrowth. Though biochemical tests are now demonstrated in this study to prove the oxygen requirement of this bacteria, historically, these mesophilic microbes have been considered strict aerobes.
Bacillus species often feed on hydrocarbon contaminants which are presently abundant in an environment like the soil near the Pasig River. Bacteria present in the soil is the most likely related to the number of pollutants because of their source of nutrients in order to survive. The water current, on the other side, is also correlated with the presence of microbes in an area. The Bacillus subtilis species attach themselves to the soil to feed on hydrocarbons found adhered to the soil. Since oil is insoluble in water, oil from the water surface attaches to the soil, making way for the bacterial growth. For the conclusion, in this study, 2 bacterial strains were isolated from long-term petroleum-contaminated soil from the Pandacan Oil Depot and from minor spills from boats passing by. The isolates were identified as Bacillus species based on their morphological characteristics. Bacteria found in oil-contaminated soil can grow in nutrient agar during isolation. Furthering the methodology used, additional number of trials, reliable colony counters, and to perform biochemical tests in order to yield significant results.